Next presentation will come right here from Nashville, Tennessee, Jon Kropski from Vanderbilt University. And Jonathan will continue on the genes that have been associated with pulmonary fibrosis and discuss a bunch of novel genes, as well as some of the ones that have come out of this center. Jonathan.
Thank you, and those are two very difficult people for me to follow, since these are who I've learned about this from over the time that I've been doing this. But I'm going to talk about something that is rather different than anything I've presented before today, to give hopefully the group some insight into some new directions that we're interested in as well. If my slides will advance. Okay, those are my disclosures. I won't beleaguer the point that has been made by many others that we've been very interested in studying families with this disease, because we think that by studying families with disease, we can identify genetic mutations that can give us a lot of insight into disease pathogenesis. I do want to note that pulmonary fibrosis, in addition to the short telomere syndromes and dyskeratosis congenita, is a feature of other systemic disorders like Hermansky-Pudlak syndrome, neurofibromatosis, hereditary fibrosing poikiloderma, and a handful of others. So pulmonary fibrosis is a feature of other systemic diseases, and I think that this concept of thinking of it as not just an isolated lung disease is probably one we're going to continue to grow through the coming years. Dr. Lloyd and Dr. Manios and Garcia have already given us a nice overview of really what's gone on through the history of this, but the point I'd really like to make is that progress really has accelerated dramatically through the course of the last several years. It took 50 years for us to identify the first genetic causes of pulmonary fibrosis, and in just the last handful, we've now got dozens of IPF susceptibility loci and common genes, and have implicated mutations in over 10 genes, evolving through different technologies, starting with candidate gene studies and linkage and genome-wide association studies, and then next-generation sequencing studies focusing on the protein coding region, and as we're going to hear more about next from Brian Yaspin, starting to expand the genetic search space using technologies such as whole genome sequencing. So others have shown similar data to this. These are data from our center and compiled from the literature to some degree. Telomere pathway mutations are the most prevalent cause of disease that has been identified to date in these families, and it's around 20% of families, and the surfactant mutations are an important cause, and as Dr. Lloyd noted, they're also relatively rare and have a somewhat unique phenotype. So what are the challenges? One question that we think about a lot is, well, why haven't we found more? And there's a number of things that play into that, including what has become increasingly clear is that this really is a heterogeneous disease at a genetic basis. This is not like cystic fibrosis. This is not like pulmonary hypertension, where mutations in BMPR2 are responsible for the vast majority of families. There's at least, as far as we can tell, there's not one other gene sitting out there that's mutated in 25% of families. We in all likelihood would have found it by now. And so what we've learned is that if you want to do linkage studies in families to really prove genetic association of a mutation with disease, you really need large families to do that, and you really need 6 or 7 affected people, and fortunately there aren't too many families that have so many affected people, although the ones that there are, we've really learned a lot from. Excuse me. And in contrast, using statistical genetics to associate the prevalence of variants in a given gene with disease, when you have a lot of different genes that appear to be associated with disease, you need a lot of people to do that. And so I think that as we move forward, we're going to really need to build larger and larger cohorts collaboratively and analyze data in conserved ways, so that we're really going to be able to make further progress. And there have been some technological barriers. Genome-wide association studies provide really interesting data about susceptibility loci, but they don't find rare things. That's not what they're made for, and rare mutations are not going to be detected by those. And whole exome sequencing, which is the primary technique that we've been using over the last several years, focuses on the protein coding region of the genome, which is a very small proportion of it, and there's a huge amount of genetic space through which risk could be conferred in families that we just haven't been able to find yet. So what we've been thinking is of ways that we can first leverage existing samples and technology to really enhance our discovery efforts. And there's a few ways that we've begun to do that, and I'm going to give you a couple of examples of these. One is by understanding subphenotypes within disease more precisely, and then more narrowly focusing on enriched groups of patients and families that have particular similar features as ones that are much more likely to have genetic variants in the same gene or a closely related gene. Another approach that we and others have used is then building upon the largest and most genetically informative families to be able to find these genes. And then we've also been moving towards using novel and innovative bioinformatic approaches to try to piece together how variants in different genes may actually all point us towards conserved mechanisms. And then as I alluded to, I think that we're reaching the time that we're going to be able to expand the genetic search region to a whole new scale through whole genome sequencing. So there are a number of subphenotypes of this disease that have been identified, and I want to highlight work from Susan Stanley and Dr. Armanios' group that came out last year, studying families with this combined pulmonary fibrosis and emphysema phenotype. And they did some very nice work that showed that mutations, in this ribosomal biogenesis factor called NAF1 were found in a number of these families with this particular emphysema and fibrosis phenotype. So the idea is if you find families that are similar, you can increase your odds of finding common pathways. We've been doing a similar approach with a particular large family that has a unique radiographic phenotype. These people in this family often get very early onset disease, and they have this apical predominant cystic pathology, which I'm not sure projects incredibly well, and cystic changes on biopsy that's not typical of UIP and not really typical of any radiographic pattern. They get disease very, very early. Several patients from these families have been transplanted in their 20s and 30s, and there's a diversity of histopathology and a diversity of radiographic features in them. So what we've done using whole exome sequencing and then subsequent candidate Sanger resequencing in these families is found that this family has a mutation in a centromere protein, the centromere protein N, and all of the affected people in this family have this same glutamate to glutamine substitution at position 46. So when we saw this, we said, well, first of all, what are centromeres? We haven't thought about those in some time. So for those who remember back to perhaps their high school or college biology courses, centromeres are these central regions of the chromosome, and they're involved in a variety of processes, in particular including aligning chromatin, during rounds of DNA replication. So centromere protein N is what's called a linker centromere, which is involved in binding this component called the inner kinetochore to the inner centromere, and CENP-N is important for allowing these outer centromere components to communicate with centromere protein A. And this particular mutation is one that's predicted to destabilize that interaction. And it turns out that these centromere components, if they're not existing as a part of this complex, they're not stable. They get rapidly degraded. So what we expected is that patients from this family, if these mutations were destabilizing these complexes, we would have reduced levels of this particular protein. So we took primary cells from patients in these family and control, and this is, my pointer's not working. The top left portion of the screen here, the red dots are staining for this centromere protein in fibroblasts from healthy control individuals compared to subjects with this particular mutation. And the nuclear levels of this were significantly reduced. So by reducing the levels of this protein, the nucleus, we expected that would reduce the ability of cells to rapidly progress through the cell cycle. So we took immortalized lymphocytes, which are a really rapidly proliferating cell type, and showed that there's a substantial increase in the non-dividing, the sub-G1 phase cells, suggesting that this particular mutation in a whole new cellular component that we had never previously linked to pulmonary fibrosis is leading to some sort of an effect on proliferative or reparative capacity. So we have a lot more work to do to try to understand this, but I think this is a new gene and a new pathway that's going to really give us new angles and ideas as to how we're going to approach this disease. Another large family that we've been studying, again with at least 7 different affected individuals, we found that this particular family has a mutation in an uncharacterized orphan cell surface receptor that essentially nothing has been known about. What's very exciting is it turns out this is really highly upregulated in the IPF lung epithelium, and its expression is substantially increased in the IPF lung. So we've cloned this mutation, transfected into cells, and shown that it appears to affect epithelial cell phenotypes, and that this is sort of a whole new, another promising pathway that's not related to telomeres that we're going to be starting to explore as we go forward. Finally, we've taken some of our existing genetic data and said, okay, for the families and the individuals that we don't know what the genes are yet, can we say what do these people frequently have variants of uncertain significance in? And so we enriched for all segregating rare variants that would be predicted to affect the protein in these families, and found that a large proportion of them have rare variants that are related to DNA repair or cell cycle signaling, which may be another way of getting that. This may lead us back to what we think is related to the fundamental biology of the telomere pathway mutations, where they lead to activation of DNA damage signaling or premature senescence, and it may be that this is really more of a combinatorial multiple hit mechanism in a number of these families. So there's clearly more. There may be exonic variation in genes that we haven't found yet, or there may be mutations in regulatory regions of known genes. I would have thought that telomerase, that you might have promoter mutations that lead to loss of function of telomerase in some of these families. We've not found that yet, but that seems the sort of thing that we need to look for. There's a whole new understanding of the complexity of how the genome is organized, and there are whole new classes of types of genetic variation that our previous genetic discovery technologies were not equipped to identify at all. These things called structural variants, which are where portions of the chromosome gets duplicated or gets flipped around and stuck back into it, and our current sequencing technologies have very limited ability to detect that. So as we are evolving sequencing technologies, we're going to be able to analyze for the presence of these structural variants as well. And as we sequence more people and more families, we'll have the opportunity to start to explore more complex inheritance patterns, digenic or multigenic pathways that will hopefully be able to help us segregate different sort of flavors of disease into ones driven by particular different driver genetic variants, kind of akin to the cancer model. So our objectives are really to leverage genetics and genetic discovery to identify mutations, to get us at the fundamental cellular mechanisms that are driving disease in those families, and then translate those hopefully to treatments for patients down the road. And I want to close by alluding first a little bit to the same paper that Dr. Armanios had alluded to a moment ago, and I agree with her that we should not be treating patients with short telomeres with androgens. I highlight this as a conceptual example of the sorts of things that we are on the cusp of being able to do. And similarly, these are data from our lab studying surfactant protein mutations. We've shown that if you use a molecule that can help accelerate trafficking or correct trafficking of a misfolded surfactant protein, you can protect against experimental fibrosis. So this is kind of analogous to what some of the cystic fibrosis traffickers like Lumacaftor are attempting to do. So with that, I'd like to thank, in particular, all of the patients and families. Truly, hundreds and hundreds of families and thousands of people who have been instrumental in helping us do these studies. As Justin alluded to a minute ago, we have a great team who helps make this happen, and I'd also like to, in particular, thank the Pulmonary Fibrosis Foundation as some of their support through a Young Investigator Award has helped drive this research in our group. With that, I'd like to say thank you.